Welcome, dear students. In chapter 4, today we need to learn more about different types of the T cells, and starting with the natural killers or the T cells. Let's start our lesson today. Cytotoxic T cells or killer T cells attacking to the foreign cells. Okay, they have a sharp function here, and after that it kills carcinogenic cells. Okay, carcinogenic is means the cell that may do you know that sometimes we suspect that some cells they are infected with the cancer. Okay, carcinogenic cells, they are a kind of cell that they are liable, or they are very weak to be cancerous cells. Okay, and sometimes they are already cancerous cells. So these cells, okay, which have ability to kill the carcinogenic cells, okay, and the transplanted organs, okay, and the body cells infected with the virus. So the attacking here for what? For the T killer cells. Also we have what? We have a kind of T cells which is called suppressor. Called what? Suppressor T cells. They regulate what? The degree of the immune response required to limit and also the discourage or inhibit the action of T cells and B cells after elimination the pathogen. So we need here a kind of manager, or we need some type of activation to give us a limited attacking. And this limiting in the attacking, or this regulation, it happens by what? By this type of T cells. Natural killer cells. As we going to continue from the last episode, as we said, we have a different types of T cells, like the killer one, the suppressor, the helper, and so on. Right now we're going to study the last type of the T cells, which is called natural killer cells. They are from about 5% to 10%, so they are very little inside your body, but they have a great function of lymphocytes in the blood, and they are produced and mature in the bone marrow. The other white cells, other white blood cells inside our body, such as what? Like basal cells, or they are called basophils. Also we have acidic cells, or eosinophils. We have also neutral cells, or neutrophils. They are what? They are distinguished, or they are differentiated from their size and color or granules, and going to know right now. They are appeared inside by using what? The microscope. We cannot observe them by just a light microscope. What about the functions of these granules inside the white blood cells? Well, the main role in the disintegration or destroying of the pathogens of the cells that attack the body, and also they ingest and digest the pathogen. Digest means they are already digested them. They are kind of phagocytosis. Phago means to engulf something, okay, and cytosis is mean to lyse it, okay. And also they struggle the infection, especially the bacterial infection and inflammations, and don't forget it. They also still in the blood circulation, okay, inside your blood for a time, for a relative short period, that this time that ranging from several hours to several days. But we have also the single core monocyte cells. We have what? Single core monocyte cells. they destroy the foreign bodies. Its main function for what? For destroying the foreign bodies and also changing into the phagocyte cells when they are needed. So they have a different function. Finally, doing what? They engulf. They what? Engulf the foreign organism. Engulf here means, do you know when you are going to take something rapidly inside your mouth cavity? This is the meaning of engulf something. Okay. This is a different types of the white blood cells. Okay, and as we said, we classify them or we divide them according to what? Their granules. Like we have neutrophil, eosinophil, basophil, monocyte, and lymphocyte, and each one of them has an important role of attacking and engulfing the pathogen or any virus or bacteria that invade your body. And here we have the macrophage. As we're going to start the fourthly, we're going to study what? The macrophage, which includes two main types, the fixed macrophages and the movable or mobile macrophages. Okay, well, after that, as we said, we're going to study what? The macrophages. They include what did we say? Two types, the fixed one and the movable one. Number one, the fixed macrophages. Actually, they are large phagocyte cells. They are what? Large phagocyte cells, okay? I mean, relative to the other cells, okay? Their types and names, depending on what the tissue where they are exist. Like what? For example, the location found in most body tissue, so they are spread all over your body. What about the function of the fixed macrophages with the first type? They are found what? One, they engulf the foreign bacterial cells. Okay, as we said, engulfing it means they are surround the foreign body and they destroy them. Okay, they aren't appear again. This is the engulfing, such as what? Microorganisms. Okay, so they have because as we said, they are large. Okay, so they have ability to engulf what? Macroorganisms, even they are entered inside your body. What the second type of the macrophages, which is what? The mobile or the movable macrophages. Their functions, they offer the information. Okay, they look like what? Like archive in your body. They are the kind of a database inside your body for offering the information they are needed and collecting about the enemies, about the microbes and foreign particles inside your body. Okay, as we said, T helper, they are activate some cells, okay, to activate the other cells. And we have another type, they collect and gathering the information for the enemy or for the foreign particles inside your body. They are called what? The mobile or movable macrophages. And also they pass information to the specialized immune system. After collecting information, we need to kill them. So doing what? They passing the information for a specific type of the soldier or specific type of the immune system or lymph to destroy it inside, found in the lymph nodes and scattered in the different body parts. And also the immune cells prepare suitable defense mechanisms such as what? Like antibodies, like specific type of killer cells that deal and treat with these microbes. So we have the other type of the macrophage, which act as an archive inside your body or the database. They offer information and collect information about the microbes and foreign bodies and pass this information for the specialized immune cells or the specialized immune system inside your body. For what? For killing or for dealing with microbes. Assisting chemical substances. After that, we're going to, fifthly, we're going to talk about some chemical substances inside our body. They have important role, okay, as a kind of activating also or the work parallel to the immune system, okay, but they are chemical substances secreted inside our body, such as what? Well, their function, they help and cooperate the specialized mechanism of the immune system. There are many chemicals. For example, we have something called, which is what, like chemokines. They are recruit or guide the migration of what, of a large circulating phagocyte cells. So they have an important role of migration of these cells to treat or to deal with the pathogen. And also we have their location and function. They are found in the blood with a large number of sites of existence of microbes or foreign particles to what? Yes, to prevent the reproduction and their spreading inside your body. If they are microorganisms and they have ability to reproduce and to spread inside your body. Also, we have something called interleukins. They are also a chemical substance inside our body. Their function, they mediate the communication or the link, the communication between the different immune cells. So they are work as what? The second type, which is interleukins. Inter here means between, okay, and leukins, it mean interleukins, it means that they are collect or linked the immune system, as we said, because the immune system, it's a scattered system. And how can we collect it together? How we need to the different parts of the immune system, they want to reconnect together by helping of interleukins inside your body, and also between immune system and different body parts. After that, they help the immune system to perform its defense mechanism or its defense function. Yet we have also something, they are complete the function of the chemical substances or assisting chemical substances. They are called what? They are complements. They just different types of proteins and enzymes inside your body, and they have also important functions, such as what? Their function that destroy the microbes in the blood after their conjugation with the antibodies. After the connecting and attacking by the antibody, they have ability what? To destroy these microbes, and also they have ability for lysing. They're doing what? Yes, they lyses the membranes of the antigens. Lysis means they are destroyed, disappear it, and dissolve their content, their components, which make them easily engulfed by the phagocytes. So the main function of the complements, they make the function of the phagocytes of engulfing. It will be more easy by the help of the complements, which is assisting chemical substances inside your body. Also, we have the interferons. They are different types of proteins that what we need to know their formation and their function. Well, starting with the formation, produced and secreted by T lymphocytes, the large phagocytes and cells invaded by viruses. What about the function of the interferons? Well, the function of the interferons, they aren't specific for a certain virus. I mean, they aren't specialized to kill a group of viruses, but they bind or combine. Yes, they bind or to healthy cells, neighboring, surrounding to the infected cells to do what? And induce them, they give them what? A kind of sparking, they activate them, okay? To produce enzymes and substances that inhibit the action of replication enzymes of the virus. So the interferons, they are a group of proteins, okay? They have an important role in what? The binding to the healthy cells, neighboring around them and surrounding them, and they activate them what? For inhibiting the replication of enzymes by producing substances. And also after that, they prevent the virus from reproduction. We need to stop the reproduction of the virus to protect the healthy cells or the healthy surrounding and neighboring cells in the around the surrounding the infected tissue, spreading in the body. Also, we have number 6, the antibodies, and we're going to study antibodies in details, and we have a lot of information about the antibodies, where are they located and how they are look like, and what is the function of them, and also you need to know the shape and the structure of the antibodies. Starting with what does it mean, antibodies? Well, the surface of bacteria cells that invade body tissue. Okay, you have a bacteria, and these bacterial cells will attack your body. Okay, suppose that, and after that they invade body tissue. They have compounds. They are called antigens. Well, bacterial cells that they have something called what? Antigens. Remember that well. After that, the receptors that found on the surface of the B lymphocytes in your body, okay, they're a group of receptors that they are spread over the cells of your B lymphocyte. They doing what? They want to collect some information, so they recognize and join with the antigens on the surface of the bacterial cells or foreign bodies. We have here antigens. They are related to it, the bacterial cells. B lymphocytes, they want to recognize these antigens. Okay, why? To let the to let the immune system see these and to observe this type of invasion with antigen on the surface of the bacterial cells or foreign bodies, and after that they produce antibodies. So antigens, they are related to the bacterial cells, while antibodies, they are related to the B lymphocytes for recognizing the antigens. And this is the types of the antibodies that we have, such as what? IgG, IgA, IgE, and D, and the other picture to show us how they are recognized. Okay, the antigens of the bacterial cells. But the question here is that antibodies also, they are specific proteins. They are known as what? As we said in the picture, they are immunoglobulin. Ig here it's short for immunoglobulin. Okay, there are 5 types: IgG, okay, or the immunoglobulin, and G and M, D, E, and immunoglobulin what? A. And each type has its own function. Okay, what about the main function of the antibodies? Well, the function of the antibodies, they circulate in the blood, okay, and lymph, doing what? The antibodies and complements. Do you remember that complements, as we said, they are a group of what? They are assisting chemical substances inside your body. Well, the antibodies and the complements, they adhere. Adhere here, it's meant to attack, okay, to stick with the bacterial cells, to offer them. What does it mean to offer them? To expose them, okay, to let them very clear for what? To the white blood cells, to engulf them, okay, or even if there is no observing for the antigen or for the bacterial cells, there is no engulfing and there is no any destroying for these pathogens. So the antibodies, okay, that they are made up, or the 5 types, and the complements, they adhere and stick with the bacterial cells, and they offer, they subjected the the bacterial cells to the white blood cells for engulfment or to destroy them. Well, but the question here is, what would happen when the B lymphocytes join with the antigens for the first time? Well, this is one of the most important questions in your lesson here. What gonna be happen? Let's show that. Number 1, they actually divide many times to produce groups of cells. Each group produce specific type, definite type of antibodies against what? Against a specific type of antigen. Again, They divide many times, produce a group of cells, each group of cells producing what? A specific type of antibodies for each antigen. Okay, so each antigen has its own antibody and so on. But what else? The B lymphocytes can invade the antigen, and the other foreign molecules can also invade them. After that, we need to study the shape and the structure of the antibodies. And from here, I need you to focus and to bring paper and pen, and I need you just to concentrate in this part of the lesson, okay? The shape and the structure of the antibodies. One, antibodies are proteins, okay, as we said. They are called what? Immunoglobulins or IgS. They need to know the shape, location, formation, structure. We need to know what? Again, shape, location, formation, and structure. Number one, the shape of the antibodies. They look like Y-shaped. What about the location? The location, they present in the blood and the other body fluids of the human and the other vertebrates. So they aren't found all inside our body, but they also found in most of the higher vertebrates. What about the formation? They are formed in which part of the inside your body? One, produced by antibody secreting plasma B cells. The structure, the antibody consists of two pairs of polypeptide chain. Do you remember the polypeptide chain? Okay. Number two, the two of these chains, the two polypeptide chains, are long and they are called heavy chains. I have a picture for you. Don't worry about the imagination here. The last piece of information about the structure here is what? The other two chains are short and they are called what? The light chains. So we have the heavy chains and we have light chains. The four chains are what? Joined together by disulfide bonds. And after that, each antibody has two identical antigens. The binding site and the shape of this site is different from an antibody to another. So it doesn't fix its shape. It changes from what? From one to another antibody. And also, the binding between the antigen and its specific antibody, at this site, it looks like the lock and its key. Get the meaning here? The binding between the antigen and the specific antibody. Antigen, again, as we said, it's related to what? To the bacterial cells or the pathogen. While the antibody, it's something from your body, okay? The lymphocytes. At the sites, they look like what? They are linked together like the lock and its key. This binding forms the antigen-antibody complex, and this antigen-antibody complex, it's a very important for your body. For doing what? The antigen binding sites are located at the two tips of the Y-shaped of the molecule, and they are known as what? They are known as what? The variable region, or V, because the shape varies from antibody to another, okay? So it doesn't fix its shape. While the rest of the antibody, the remainder of the antibody, the other part, is known as what? The constant. Again, the antibody, it consists of two main parts. One of them is variable, we call it V shape, and the other is constant, and it doesn't change, and we call it C, or the constant region. Because what? Because it has a constant shape and the structure in all types of antibodies, contradictory to the V shape or the V region, which is a variable from antibody to another antibody.